All right, so we are going to start our afternoon session, and the first talk will be by Dr. Ridlen's students. So can you please introduce them?
Yes, absolutely. Hello, I'm Jenna Ridlen. I'm with the biology department. Thank you all so much for being here. So these are two of my excellent students that I had the pleasure of having in my bioinformatics class last semester. And they have both of them decided to go on and do independent research with me this semester, and will be presenting today and then also at the Alabama Academy of Science conference here in a few weeks. So this is Alyssa Jones and Hunter Ballard, two of our rockstar students from the biology department, and they're going to be talking about a genetic variant in Treacher syndrome, which is micronathia, or like a small chin. So.
You can't see it all the way up there.
Yeah.
There we go.
Here we go. Like she said, my name is Hunter Ballard, and this is Alyssa Jones, and this is our presentation on TCOF1, which is a gene that affects the disease Treacher syndrome.
So a little bit about Treacher syndrome. Treacher syndrome is a genetic disease that is pretty much always going to be passed down from a parent, typically a mother for the most part. Treacher syndrome is mainly because of three specific genes, POLR1C and POLR1D, but the most common one is going to be TCOF1, which is why we decided to go with that one to study. So Treacher has multiple effects. While it doesn't typically affect anything with intellect, it does cause deafness, blindness, and as you can see up here on the top right, it can cause specifically, one of the most common is going to be micronathia of the lower jaw, so a smaller lower jaw, downward slanted eyes, a cleft palate, and downward slanted ears.
So our objectives with this research were to determine the relationships of multiple single nucleotide variations within the gene TCOF1 to determine A, the pathogenicity of a variant at these specific amino acid sites, and B, establish any trends or relationships between these to determine if there's some further that we can investigate as far as treatments or cures goes in the genetic domain. The materials and the methods that we used are some really good public databases along with a protein modeling software. The databases include, or our tools include a CAD score, which is a combined annotation dependent deletion. It's a tool that determines the likely pathogenicity of a mutation at an amino acid site. Next we have polyphen 2, which is another form of CAD score. You can see it's these two sort of spectrum looking charts at the top that goes from green to red. That's another form to determine the pathogenicity, and it's sort of double source. There's something called a humdiv and a humbar, which is sort of like a spell check for the CAD score. Next, we used Yasara, which is the protein modeling software. You can see that right below. It's the little shapes and things. We obtained the genetic, the protein information from a website called AlphaFold, which is then we uploaded it using a TDB file, which gave us this large protein structure that we were able to single in and find our exact locations of our both wild type and variant amino acids. We also used the multiple sequence alignment tool on UniProt, which is at the very bottom there. It's all the letters that go across. And that's just a tool that determines how conserved these amino acids are across multiple species. So it's the humans and chickens, and so on and so forth.
Okay, so he talked a little bit about the CAD score. The CAD score is what determines the pathogenicity of the gene, or of the variant that we're looking at within the gene. So as you can see here, this is a scatter plot that we used, making Excel, of all of the different variants of clinical significance. Of course, there were more, but we varied them out a little bit so it only shows the misses, mutations, and things like that. So you can see the ones that we have highlighted in purple up here at the top, 621. This is at position 621 with a CAD score of 3.495, which is pretty high, meaning that this is going to be a probably damaging gene, which you can see if you'll scroll down just a little bit, Hunter, so you can see. This one right here, it is possibly damaging. And then our other gene over here, excuse the technical little workarounds, at the 39th position has a CAD score of 0.972, which is actually very low, and surprisingly, it was benign, which you don't find much of with Treacher Collins.
Yeah. So moving right along from that, these next tables that you can see, the rows with all the different colors, this is a demonstration of the raw data in an Excel sheet. And what's interesting about this is our two variants, even though they're surrounded by different levels of pathogenicity. So the one right here on the left is the one that we believe is likely benign with a low CAD score, and it's surrounded by multiple pathogenic variants. While the one over here on the right, it's cut off a little bit too, but it's surrounded by several benign and several unknown variants, although the CAD score is rather high for it, and it's likely pathogenic. It's just an interesting trick.
Going back full screen here. So looking a little bit at our structures that we used, it was very difficult actually to find a PDB file for this. Most of the time you'll find PDB files using something like the Polyfin or the NCBI website. But what we had to do was we dug and dug and found one on AlphaFold and used Yasara to put that structure in like an artificial reality application so that we can manipulate it and change it however we like. So as you can see here, this is our gene that was benign. This was phenylalanine at the 39th position, you can see here. This is what it looks like highlighted in red with the side chain highlighted as well, the amino acid side chain. And over here on the right, this is when it was mutated to leucine at the 39th position. You can see it significantly changes the amino acid side chain, but it does not cause Treacher Collins. Now on the right here, this is at the 621 position. On the left, you have glutamic acid. Glutamic acid, you can see, is a rather long side chain, and then when it's mutated to valine, it turns it pretty short, and something in this is causing Treacher Collins.
Right below that, I referenced it earlier, is what's called multiple sequence alignment. So all these letters that you see across here are amino acids at various positions within a protein across different species. So you can see top to bottom right here, the species are, you have an African clawed frog, a mouse, a horse, a chimpanzee, human, a central bearded dragon, and then the last one right here at the bottom, which is comes into a bit of interest, is the chicken. On this first one right here, this represents our benign variant, which is represented uniformly on the left side of the screen right here. And we believe it to be benign. So it's not necessarily as intriguing as our bottom one that we'll get to. But what it is intriguing is that while our wild-type amino acid is conserved across every other species, chickens are an exception to this. So at the 39th position for the species of chicken, they don't have a phenylalanine, instead they have a leucine, which is something that would be interesting to investigate as to what effect that has on the overall development of Treacher Collins as far as variations go. And then at the bottom, we have the same thing before our second mutation at location 621. This is where the glutamic acid becomes the valine, but right here you can see our wild type glutamic acid. And it's not conserved in both the central bearded dragon as well as the chicken. But what's interesting here is that it's a misalignment on the actual protein. So there's not actually any amino acid right here in the way that it lines up, which is something else that we could delve deeper into with further investigation.
So, in conclusion, the main things that we found was that the SNPs at the F39L position and the E621B position, although they are both point mutations within the TCOF1 gene, they're very different. The F39L is not very pathogenic when it comes to causing Treacher Collins, but E621B was.
Moreover, the specific structure changes seem to play more of a role in determining the pathogenicity of a variant rather than the position of the amino acid within the chain. So that's something I was referencing earlier with the multiple sequence alignment. We could spend more time investigating species like the chicken or the central bearded dragon to see how mutations at different locations affect Treacher Collins syndrome more. And that would be something worth investigating to see if there's any possible treatments or cures that we can come up with for the disease at the genetic level.
Overall, Treacher Collins remains to be a very, very heavily understudied disease that would need a lot more advanced technology than what we have access to at this time to be able to actually learn more. And you can see our references up here. These are all the tools that we used, and we wanted to give particular gratitude to the JSU Department of Biology for helping us do this, and Ms. Michelle Morris with the Hudson Alpha Code Project, which is a big part of what this presentation is. And then special acknowledgements to Dr. Jenna Ridlen, who advised us throughout this entire study and helped us out throughout the process.
Thank you all.
Are there any questions?
Yeah, any questions for our speakers?
So in humans who have Treacher Collins, are there chromosomal abnormalities that are associated with it, or are there just like these random point mutations of unknowns?
What we've noticed so far is mainly the point mutations of unknown significance. You can see it if you actually look at cases, especially in familial cases. It can turn out completely different. There's going to be different levels of effects. So the reason I actually wanted to do this is because in my hometown there is a family, and all four of them, the mother and then three daughters, all four of them have Treacher Collins. But if you look at them, it affects them in completely different ways and levels of severity, and I found that very interesting. So we don't really think it's in the chromosomes as far as we know, but like I said, there needs to be more research done on this disease.
So do you know what if any genes they have particularly, this family?
I do not know particularly.
They might not know either. 'cause you
Yeah,
have to
there's-
pay for genetic
There's no
counseling.
telling, but
Likely
I'm not, but it's most likely TCOF1.
Maybe
In the study, nearly all cases were caused by TCOF1. There was very
81
little.
to 93% to be exact.
Any other questions? No? I have one question. So in your presentation, you talked about CAD score. So could you please explain it one more time?
Yeah. So the CAT score is, it's almost like just an independent tool of its own. I'm not sure the exact formula that they use to determine this, but they look at things like the structure changes as well as the position, even though we determine the position is not necessarily has.
So structure means volume?
Yeah, so the actual like structure, when a amino acid is changed, like right here, you can see how different it is from
Okay.
its original wild type. Usually is a pretty good indicator of how bad it's going to be. So the CAT score sort of takes that into account and produces a numerical score to determine it's going to be really bad, is it going to be just a little bad. And so you can see right here, like this is a very large structure, and we lose a couple of sections when we change our initial one for F39L, but it still remains a pretty low CAT score, so it's not likely to be true. And then over here on the second one, we lose what we're here to be like several more or more important sections of our structure, and so that's why the CAT score will be higher for that one.
All right, thank you.
Dr. Raymond, you have-
It's a weird thought.
okay.
We're talking about the DNA, changing the protein structure, and this is all modeling that change in protein structure. And I know that I've seen several of these talks throughout, and it's all been kind of like, well, we see this modeling, and then we don't know exactly what's going on down the road, some features within that. And I don't know, it just struck me that it might, whether there's any modeling for prions, like mad cow disease, where you're causing, this mutation causes a change in this protein that actually causes it to misform whole other proteins. And I don't know if there's a program, you've been looking at the programs, that's what I was thinking, if there's a program that could predict that, that would be another chain in the link.
There honestly most likely is something, and I feel like we definitely covered something that sounds similar to that last semester in class, but it's not something that we dove into with this. But if you looked into it more, and to find the right files and the right genetic sequences to be able to properly look at it, then you probably could do something like that.
Yeah, this is all a pretty new field, so it's difficult to come across information. But Yasara has several tools, or protein modeling has several tools, and it might still be in a developmental phase where it's showing us single amino acid changes, but it hasn't yet shown us, the effect on secondary or tertiary structure of the proteins.
Oh, I thought it was making predictions on that, it's not doing that yet?
It is, but it's just at a smaller scale, so it's really just viewing them.
Static. It's making static predictions.
Oh,
It's
okay.
not- You're talking about dynamic.
Yeah, I was talking about three-dimensional dynamic, I thought that's what the CADs will include.
No, that's,
Unfortunately, no. It's kind of, it's
what-
not down to an exact, we can't, it's not 100%. I mean, it's gonna be hard to do 100% when this is an alternate reality kind of thing and not an actual person just that we're studying, but.
All right.
Oh, sorry.
I'll get set up while I'm answering that
Yeah,
question.
if no more questions, let's thank our speakers.
Thank you very much.
Dude, I'm not even trying to.